So our next speaker is Christian Rutz. I hope I'm pronouncing this right. He's from the University of St. Andrews, UK, and he's going to present us the strange framework for improving experimental designs, reporting standards, and reproducibility in animal behavior research. So we're waiting for Christian to join us. Hi, Christian. Can you hear me?
Yes, I can hear you.
Yeah, you can switch your camera and mic on and share your presentation now.
Okay, I'll go into full screen.
Yeah, it works. The floor is yours.
Thank you very much. Thanks to the organizing committee for Animal Behavior Live. This is a really fantastic conference, and thanks for everybody who joins for the talk. I would like to introduce the STRANGE framework today, which was designed for improving experimental designs, reporting standards, and reproducibility in animal behavior research. And before I start, I would like to acknowledge my co-authors on this presentation, Alice Langley, Kat Hobeta, and Mike Webster. We are all based at the University of St. Andrews. The STRANGE framework was introduced last year. It's a relatively new idea, and it's a framework that was designed to help animal behavior researchers identify, mitigate, and report sampling biases. It applies to all taxa, vertebrate as well as invertebrate, and across all study types. So that's lab and field studies, experimental and observational work. It was inspired by the highly successful WEIRD concept in human experimental psychology many of you will have heard of, and I will unpack the STRANGE acronym in a second. It provides a useful memory aid, and I would like to encourage you to routinely ask yourself, how strange are my study animals? And that should make more sense in a second. We as animal behavior researchers, we collect data for samples of animal subjects. We usually don't have the resources, and it's logistically not feasible to study the entire population. But the aim is to then use data from these samples to draw inferences about the wider population. And a problem with that is that very often the samples we draw are severely biased. So you can see in the schematic, my sample includes 3 of the orange crows, which really isn't representative of the wider population, which mainly consists of gray crows. And those biases could refer to an imbalanced sex ratio. Maybe those 3 birds have a particular personality type or body condition. These are very common biases. Biased sample composition can impact the interpretation of study results, limit the generalizability of findings, complicate comparisons between studies, and ultimately hamper reproducibility. And this actually seems to be a well-known issue within our research community, but it's not really systematically tackled. It's the kind of conversations we have at conferences and workshops, but these concerns rarely find their way into our study designs, let alone our publications. The STRANGE framework was designed to address this problem head-on. I would now like to unpack the acronym for you. Every one of the 7 letters stands for something. When we designed this framework, we were thinking about what are the factors that drive variation in animal behavior. And of course, that's the animal's genetic makeup, its social background and experiences, opportunities for social learning, and its individual experience, things it may have acquired through trial and error learning. So those are the 3 main building blocks. But we then complemented that with other sources or drivers of variation or biases. One of them, an important one, is trapability and self-selection. So how likely it is that a particular demographic group ends up in my test sample, and I will explain that more. The subject's rearing history, what it has experienced in terms of enrichment or throughout its life, previous experimental exposure, acclimation and habituation, and natural changes in responsiveness that can occur throughout the day or with seasonal cycles or as an animal matures. And those 7 factors make up the acronym STRANGE. They are not in themselves problematic. They are actually very often the focus of well-designed research projects, or they are confounds that are explicitly controlled for in statistical analyses. But concerns arise whenever samples are biased with regards to any of these 7 factors, and researchers do not account for this. I just want to give you some examples that these issues really exist and that they matter. I believe that these strange effects are common, but usually ignored. The examples I present here are actually studies that set out to demonstrate these effects. So they haven't done anything wrong. They haven't overlooked these issues. They, in fact, try to spotlight these issues. The first one is a classic study on capuchin monkeys that used a self-selection paradigm. Those are laboratory animals, and they were allowed to self-select to participate in an experiment. And the authors found that monkeys that are less assertive in terms of their personality and more open are more likely to participate in experiments. And those that are more assertive and less open were the ones that didn't participate. So clearly, if you don't adjust your testing protocols, you end up with a biased sample. That's a fantastic example to illustrate the issue of self-selection. In this example here, that's guppies, the authors found that fish from domestic stocks swim farther than feral subjects, and activity, their swimming activity, declines with acclimation. So we've actually got 2 effects interacting here. One is it really matters what your stock of test animals is, whether they are domestic or feral. And secondly, it matters when you test them. So if you were to record your data 2 minutes into the habituation or acclimation period, you get a different result compared to if you tested them 1 day later. So good example of genetic makeup and acclimation. And here finally, as an invertebrate example, fruit flies that experienced mating previously or are tested in the light rather than the dark turn out to be more social and aggregate more closely. Again, 2 factors involved here. One is experience, mated or not mated previously, and the other is natural changes in responsiveness. So it matters whether the lights are switched on or off. In this article in which we introduced the STRANGE framework, we have a supplement that gives a very large number of studies that illustrate that these effects exist. We hope that STRANGE will improve research reproducibility in our field by affecting the whole workflow, from the conception of an idea to the publication of the data we have gathered. So at the project planning stage, we hope that researchers will consider STRANGE-related sampling biases when they design their studies and they prepare their grant proposals. So there is an opportunity to take these issues into consideration as you design your study, choose your study subjects, hone your testing protocols, and so on. Funders then can play a role in evaluating these considerations and whether these sampling biases have the potential to affect study outcomes, and if so, whether that has been acknowledged by the researcher. Then once a piece of research has been completed, it'll be submitted for publication to a scientific journal, and then reviewers and editors can evaluate the work. And we hope that researchers will start taking the STRANGE guidelines into consideration for the reporting. Ultimately, we hope this will lead to better research, enhanced transparency in how we document our work, and improve reproducibility. So this really is intended to be a push for positive change. It's not meant to be a policing tool or a prescriptive framework. It's hopefully a move in the right direction for us as a community to think about these issues. It's really exciting to see that this seems to have struck a chord, and an increasing number of journals, some specialist journals in animal behavior and some broad interest and highly selective journals have started supporting Strange, and they have integrated it into their author guidelines. I hope you see at least one of your favorite journals here. Many others are currently in an editorial consultation process. They're discussing the framework, but it's really exciting to see these early adopters and papers coming out from these journals that use the framework. So how does Strange work in practice? We have developed what we call a 3D approach. The 3D stand for design, declare, discuss. So at the design stage, when you design a new study, we would encourage you to critically evaluate the importance of the 7 factors of Strange. That's a really fun exercise to do within a research group as part of a journal club, to just talk them through and really think about how they might affect study outcomes. Consider diversifying your sample of animal subjects to make it as representative of the wider population you wish to make inferences for as possible. So, for example, you could attempt to balance the sex ratio of your subjects. Consider making adjustments to experimental protocols to reduce bias. So, for example, you could try and tweak protocols so you engage shy individuals that may otherwise not participate in a self-selection paradigm. I mentioned the monkey study as an example. Implement these design changes, but of course, as far as possible within the 3 R's considerations about animal welfare and logistical constraints. It's of course understood that the perfect study doesn't exist. But then signal awareness of these considerations and applications for ethical approval. Strange really matters for animal welfare considerations, and I'm happy to discuss that further, and for external funding. Once you have conducted your study, you can use the framework to make these declarations. The Strange framework encourages authors to provide detailed information on subject attributes. So those are some of the ones that may matter. And importantly, for many of those, you may not have data, and that's okay. It's okay to give a nil response and say, well, I don't know what the personality type of my subject is, but that is better than leaving it open and not documenting this. And the framework suggests to make these declarations for the final sample of subjects that contributed data to the study, as well as those subjects that were part of the original sample, but did not contribute data for whatever reason. So those are the excluded data points. And I think it's really important to document what these animals were they the dominant animals or the subordinate, the old or the young ones, were they the shy or the bold ones. A second part of the declaration bit is to then evaluate scope for sampling biases and describe what efforts, if any, were undertaken to mitigate potential sampling biases. So in terms of sourcing representative subjects and adjusting testing protocols. Now, really important, like once these declarations are made, it would be good to add 2 brief statements to the main text of research articles. One in the materials and methods that simply evaluates the strangeness. In many cases, it's a matter of putting a sentence in that says, well, this is pretty representative, or it's not representative because those are the subjects we had access to in our laboratory colony. And another one in the discussion, and this is important, explaining how potential biases may limit the generalizability of the reported findings. So this is where the findings are tied to the properties of the test sample. So if I conduct a study on New Caledonian crows from one particular study population, well, it's good at that stage to say, well, my findings apply to this population, but may not generalize across all New Caledonian crow populations. Strange is integrated with other frameworks, and you will be familiar with these at the design stage. It's the prepare framework for planning experimental work with animals, and of course, the 3 Rs, replacement, refinement, and reduction. At the reporting stage, it's the well-established arrive guidelines. So you can consider strange as a bolt onto arrive with issues that apply specifically to animal behavior research. In recognition of how strange is integrated into this ecosystem of existing guidelines, UKRN have just adopted strange as one of their initiatives. That's the UK Reproducibility Network. Just some take-home messages. Strange applies to actually not just behavior and cognition experiments, but physiological, biomechanics, medical and translational research, and many other fields. I've started discussing with colleagues all sorts of applications we hadn't thought of when we first came up with this. It's really important that it is pragmatic and light touch. There are no checklists you have to fill in. It's not cumbersome. Instead, the strange framework strives to empower authors to decide what applies to their study and what doesn't. It's meant to be an analysis tool. It's meant to encourage authors to pause for a second when they write a manuscript, consider these issues. And quite often, as I said, it could be a few sentences in a manuscript, it could be a supplementary table that makes all the difference for the readership in terms of understanding the findings. It's easy to implement. Just think about this design, declare, discuss approach. There are plenty of papers coming out now that use the framework, and I'm happy to share some examples if people are interested. It's also an engine for generating interesting new questions and research opportunities. So we hope that people don't think about this as another hoop to jump through, as another check whether you've done everything right. Actually, once you start thinking through the sources of biases, you very quickly come up with interesting ideas for follow-on research. That's something I've seen in journal club discussions and workshops. It has sparked a grassroots push for positive change, and I really hope that plenty more colleagues will get on board and that you will help us spread the word about this. And I really hope you will consider joining the Strange team. We are building this initiative. We are looking for help with a range of activities. I can talk more about that. And I hope you stay up to date with anything strange. We are on Twitter. Please consider following us. Thank you very much for your attention.
Thank you very much, Christian, for your talk. So while we are waiting for questions on the YouTube chat, can you hear me?
Now I can hear you, yeah.
All right, it looks like my internet connection is not good. I was asking how people could be involved with Strange.
Yeah, please get in touch with us, send us an email or reach out on Twitter. We have a range of activities we are planning. So apart from workshops and webinars and discussion groups, we are planning to develop teaching materials that can be used in undergraduate and postgraduate teaching to help train the next generation of animal behavior researchers and work together with them on identifying these problems and addressing them. We are putting together a website. We are planning to build a network of strange champions. So you could become a strange champion in your university or research institution where you host journal club discussions and try and spread the word. And we've developed some guiding questions for these. They are really good fun. So I would encourage people to either run with it or get in touch with us. There are plenty of opportunities to get involved.
All right, thank you. We have some comments and questions in the chat now. So Amr Salis, sorry, has no question, but she says, this is so great. I really like the idea. And then April Timis is saying, such a great talk. How long did it take to come up with the strange concept?
It took us not that long, actually. It was something we had, my co-author Mike Webster and I, when we put together the original article, we had been discussing this over coffee, and we realized that there is a problem. And then it was a matter of a few months to piece together the pieces of the puzzle and write the article. But the exciting thing is that now that the community is engaging with it, that framework evolves. So we never considered this to be something that is set in stone and it's being enforced onto the community or journals. We expected it to be a proposal to the community for how we might be able to change things for the better. And so this is now really exciting to hear the feedback, to hone the wording of the author guidelines. We are working together with journals who adopt this to get it right on a journal by journal basis. And we constantly get feedback that people say, well, have you considered that it applies in this context as well? And we haven't. So this is the exciting bit. And this is where we really look forward for interaction with the community.
All right, thank you. So you have also several comments from Mariela Herberstein and Franziska Fruerig that are saying that it's excellent work and very relevant subject, especially in the psychology field and ethology field. And one last question from Ebi, who is part of the ABL committee, is saying, great talk and a great framework. How do you go about deciding the level of rigidity in the guidelines to follow versus how much control researchers have? Will you modify the framework based on feedback from the community?
Yeah, so the latter point is important. We still haven't learned of anything that is missing in the acronym or the framework. So it seems to cover all the bases, but of course the nitty-gritty matters and how you use the framework. And as I just said, it's meant to be reshaped and evolve as people engage with it. And with regards to how rigid it is, not at all. It's meant to be an aid. It's meant to be helpful. It's not a prescriptive tool. It's not a policing tool. It's not meant to say, oh, you've done bad research. All your crows are young birds, you have done a bad job. No, it's not about that. It's about thinking through the underlying biology, thinking about what our findings mean, being transparent and how far we generalize. And it's meant to be fun and helpful. And hopefully in a few years' time, we can look at whether it does help reproducibility by increasing reporting standards and transparency.
Well, that's great. Thank you very much, Christian. So we now have a 30 minutes break. I invite you